Good morning, students. Today, in the last class, we had talked about the chromosomal theory of inheritance. Then we had talked about the behavior of chromosome, comparison between the behavior of chromosome and genes. So today we will talk about the next topic in this chapter, that it is the linkage and recombination. Okay, what it is? That is the linkage and recombination. Before going to the linkage and recombination, here one of the scientists whose name is T.H. Morgan. What was his name? That it is the Thomas Hunt Morgan. Thomas Hunt Morgan. He was one of the scientists whose name is T.H. Morgan. So he best known for his work on the fruit flies. He best known for his work. On what? On fruit flies. Okay. What is that? That is one of the insect. That is the fruit fly. Okay. On fruit fly. So this fruit fly, scientifically we can call it as the Drosophila melanogaster. What we can call this fruit fly? That it is Drosophila melanogaster. What was the scientific name for the fruit fly on which the T.H. Morgan has been worked? That is the Drosophila melanogaster. So he chosen the fruit flies for certain reason. Okay. So this experiment has given one of the strength to the experimental genetics. Okay, so here father of experimental genetics means that it is the T.H. Morgan. T.H. Morgan is also known as the father of experimental genetics. What he was known? He was known as the father of experimental genetics because he has worked on a particular genes of what? Of fruit flies. And the next thing here, Drosophila is one of the excellent model, genetic model, and is called as the Cinderella of genetics. Drosophila melanogaster or fruit fly, which is also known as the Cinderella of genetics. This is one of the excellent genetic model to study the inheritance or variation. Why he has chosen for this fruit fly for his work? What are the reasons? So here there are some reasons because of some following reasons. What is the reason behind choosing for what? For the fruit fly for his experiment. First of all, they can easily rear in the laboratories. They are very small in size, easily available. For that reason, we have chosen the fruit flies for his work. First reason, that is easily reared in laboratories. Easily reared in the laboratories. Easily reared means we can nourish them, they can reproduce, they can undergo into the reproduction, everything, their life cycle can be completed in the labs, that is either it may be a school lab, college lab or any scientific lab. On what? On simple culture media. On simple culture media. Culture media means which provides the nutrients for their growth. Okay, this is the first reason. Second reason, it has a very short life. The fruit flies will have a very short life, that is of 10 to 12 days. For this reason also, he has selected the fruit fly for his experiment. Okay. So this fruit fly has our Drosophila melanogaster. Fruit fly has a very short lifespan. Very short lifespan. Short lifespan means from their birth to their death. Okay, it is of very short time, that is around 9 to 10 days. Only this much is their lifespan. For this reason also, he has selected or chosen the fruit flies. Okay, then they can produce a numerous generation from their parents. Next, a single mating by pair. A single mating by pair, okay, can produce hundreds of progeny. Can produce hundreds of progeny. Progeny, that is the offspring. We can produce a numerous things, in the sense they can one mating. Okay, one mating can produce 100 number of progeny or 100 number of offspring. Okay, so by two flies there will be production of many number of offspring. For this reason also he has chosen the fruit fly for his experiment. Okay, the next sexual dimorphism is clearly evident or is clearly not. Sexual dimorphism is clearly evident. Means what? Sexual dimorphism is what? That it means we can recognize the male fly and as well as the female fly. How we can recognize it here? That the males will be the bigger one and the females will be the smaller one. Sorry, the males will be here in the sexual dimorphism. The males will be the smaller in size and the females will be the larger in size. Okay, in this way, by seeing their size, by noticing their size, we can say that this fly is male and that particular fly is female. So that character, that is the sexual dimorphism, here it is clear. In whom? That is in the fruit flies. Okay, the next, it has a 4 pair of chromosomes. How many chromosomes are there? That it has 4 pair of chromosomes. 4 pairs means how many? It will become 4 pairs. It will become 8. 8 chromosomes. See here. 1 chromosome, 2 chromosomes. 1 pair. This is the second pair. This is the third pair. This is the fourth pair. Okay. So 4 pair of chromosome means how many chromosomes will be there? 1, 2, 3, 4, 5, 6, 7, 8. 8 chromosomes will be there in each fruit fly. Okay. 3 pairs are autosomes. Among these 4 pairs, you see here, these 3 pairs are autosomes. Autosomes means what? That chromosome which is responsible for the body character. Okay, that is the autosomes. Among these 4 pairs, 3 pairs are autosome and 1 pair is allosome. Allosome means what? These are the sex chromosome which determines the sex of the particular fruit fly. Okay, allosome, 1 pair is allosome and 3 pairs are autosomes. Okay, so whatever the sex chromosome, here I will write it. See here, there will be presence of 4 pairs of chromosomes. Among 4 pairs, 3 pairs are autosomes and 1 pair is the allosome. Autosome means which is meant for the body character. Allosome means it is meant for the sex of the organism. So here the allosome in the females, what it will be? In the females, this chromosome can be represented as the X. Okay. In females, the sex chromosome or allosome will be X. But in male, it will be the X1. Okay. In female, it will be XX. And in male, it will be the X1. Okay. So, due to this chromosome, presence of the sex chromosome only, the sex of the fruit fly will be decided. Okay. For this reason, this is similar to humans also. In the human reproduction chapter, you had studied how to determine the sex of a human. Okay. In that also, the same chromosome. The female can be represented as XX chromosome and male can be represented as XY chromosome. Okay, the same thing we can see here in the fruit fly. Next, Morgan has been experimented on these fruit flies on a term that it is the linkage. First of all, what is linkage? I will say. See here, Morgan's experiment on linkage. So let's see about their experiment. See here, first of all, Morgan has been carried some of the experiment, such as how the Mendel has been crossed, such as monohybrid cross or dihybrid cross. In a similar way, Morgan also has been crossed, which cross means that it is a dihybrid cross as well as a monohybrid cross. On whom? On the fruit flies. Okay. Morgan carried out several dihybrid crosses in Drosophila melanogaster. Here, Morgan carried dihybrid crosses in Drosophila melanogaster. Drosophila melanogaster. That is the fruit fly. Okay. So, what are the characters as you had studied in pea plant for dihybrid cross? They have taken the shape of the seed and the color of the seed. Okay, that is the yellow, round and wrinkled green. Like that we had crossed. But here we want two pair of contrasting character for to carry out the dihybrid cross. Dihybrid means where we are taking the two pair of contrasting character. In a similar way, as Mendel has done in pea plant, like that only Morgan is working on what? On Drosophila melanogaster. So what are the character means? He took a wild male with the here first one. He took wild male with the two characters, that is brown body and red eye. Brown body and red eye. These are the two characters in whom? In the male. That too, this male is wild male. Okay. What are the two characters? We want two pair of contrasting characters. One is brown body, another is red eye. Here, one more contrasting character will come on top. Then he crossed this male here. He crossed this male figure. With whom he crossed? That he crossed the yellow. Okay, red eye and the white eye. So here, how we can represent it? Means yellow body means we can write it as the capital Y. White eye means we can write it as the W. How we can represent it? With the capital Y and a W. So he crossed a male with a female. Male has the brown body and red eye. Then the female has yellow body and white eye. So first we will see about the cross. So this one I will write around it and I will show the cross, how he crossed. See here, parents. First of all, two cross are there. First, I will explain the cross A. This is the cross A. First, these are the parents. Parent means one will be the male and another will be the female. Okay. So here, For better understanding, they had given such diagrams. Okay. This is the female. This is about what? This is about the female. Okay. Female where it is yellow body and white eye. Okay. See here. This is the genes or alleles for yellow body, which is represented as the capital Y. These genes are responsible for what? For the yellow body, capital Y. Because we are talking these genes or these chromosomes are of what? Of the female, where it is yellow body and one more pair of gene is there, that is white eye. W. Yellow body, white eye. Same thing, yellow body, white eye. Okay, here this one is crossed with the male with the brown body and red eye. Okay, here this is the, in bracket I will write here, this is the female with the yellow body, which can be represented by Y, and white eye, which can be represented as W. Okay, these are the two characters. Then which is crossed with the wild, means we can represent it with a plus sign. Okay, see here. Here this is crossed with the So here, this is the female and this is the male. What is this? This is the male, that wild type. Okay. Here, this male, sorry, this is the wild type male and this is the female. And wild type male will have these two characters, that is the brown body, which can be represented as Y plus, and red eye, which can be represented as W plus. Okay, here I will write, this is the male, Y plus means that it is the brown body, and W plus means that it is red. Well, a cross is made between a mutant female and a wild type male with the yellow body and white eye with the brown body and red eye. In the F1 generation, what happens? Here you see, this next one comes the F1 generation. This is the F1 generation, as Mendel has been first from the parents, then gametes, then F1 generation. So in F1 generation, what happens means, here you see, like this. Okay, so here this is yellow body, white eye, and here it is red body, red eye. Sorry, brown body, red eye. In F1 generation, the females were like this, and what about the males? The males were this is the sign of wildness, okay, in males. So here, how genes will remain, sir. Like this. Why this is going to take place is because here the chromosomes are going to combine. Okay, chromosomes. If I show, these are the chromosome. This is the another chromosome. This is the segments like this. When they cross with each other or when they are going to recombine in the next generation, the chromosomes will be like this. Like that only, here also you can see the recombination. What you can see that it is the recombination in the F1 generation. Here you can see the female will have both the combination. Okay, that it is white eye, sorry, yellow body and red eye, brown body, as well as the white eye and the red eye. But the males will have a, that it is the yellow body and white eye. In the, so what will be the F2 generation here means, what is the percentage of the recombination means, here we see in F2 generation, in a F2 generation, here, this is the Y plus and this is the W plus. This is wild type. The females now becomes the wild type in the F2 generation. Like this it will be. Okay, here W is also yellow, white. One more cross. So this will be the recombination. How much percentage is recombinant here means that is 1.3%. And how much percent is similar to the parental means that is 98.7% or 98.7% size. Then recombinant type that is 1.3%. Okay, now once again I will tell you, when Morgan crossed the yellow body female with white eye. There are two characters, yellow body and white eye. When he crossed like these females with the brown body and red eye males, that too wild type. Okay, they cannot be easily recombined. To them, we can call it as the wild type. Okay, when they crossed both the parents in the F1 generation, he found that some of the females have a character of what? Wild type, wildness. Okay, that genes you can see here because the chromosomes are going to recombine. There you can see the blue color shade, and here there is no blue color shade in the female. But in the F1 generation, the female will have a blue color shade. This means that there is something is going to combine. What is that something? That is the chromosomes or the genes that is going to recombine here. And here you see, due to the wideness, we are showing a blank column or a blank chromosome. Okay, that same chromosome will be inherited to the next generation. But what about the character? Here, this character has been exchanged to this. Okay, in the next generation. The next here, next in F2 generation, you see, and once again, if you cross in this, you can see in the F2 generation, again recombination is going to take place. This wild character will come into the female. Okay, till this time, this is present only in the males, but in F2 generation, this wildness came into the females. Yes, sir. This wildness came into the females. Then here also, some characters have been exchanged or recombined. There also you can see, and here in the male, both the characters are there. One is white body and what is that? White eye, and here that is the brown body. In F2 generation, the males have what is that is the brown body and white eye. This is one of the new character which is found in the Drosophila. Okay. The next here also the same thing you can see. So percentage of combination, how much it is means 1.3, and the percentage which is similar to their parents, that it is around 98.7%. So this is about what? About cross 1. In the next class, I will explain about the cross 2. One more class is there to explain this experiment on what? On Drosophila melanogaster. Okay, thank you.